So this presentation was originally advertised as a Hungarian language presentation, but we switched because of the international collaboration to English. Therefore, please allow me to give 2 sentences only in Hungarian for those who are expecting this presentation in Hungarian. Szóval elnézést kérek azért, hogy ezt magyar helyett angolul tartom ezt az előadást, de mivel ez egy nemzetközi együttműködés keretében megvalósult kutatás, a nemzetközi partnerekre való tekintettel az utolsó pillanatban úgy döntöttünk, hogy angolul lesz megtartva az előadás. Bízok benne, hogy az ábrákról érthető lesz a mondani való. So back to English now. I would like to present you the result of our phylogeographic research, what we did in collaboration with Czech, Polish and our Hungarian researchers, because we had a research grant that was financed by the International Visegrad Fund, and in frame of this collaboration, together with these institutes and these ladies and gentlemen, we provided a kind of phylogeographic picture and genetic research of different butterflies and moths, usually those which are of conservation concern. So in this work I would like to talk to you about the Jersey Tiger Moth and of course what are our results. So as a kind of unusual way of presenting this talk, but I need to start with some sort of acknowledgement, because we have received quite a lot of samples from all across different, mostly Visegrad countries, and yes, evidently it was a big work. So what is this target species what we were working with? So this is a moth species which belongs to this Arctine subfamily, and it is in the Noctuidae superfamily, so a kind of moth species. The unusual thing with this species, Euplagia quadripunctaria, Jersey moth, tiger moth, the English vernacular name is. So what is unusual with this species is its part diurnal activity. So although it's a moth species, it's very easy to meet the adults, of course, in daytime. Actually, it's a frequent species. In Europe, it's not a rare species at all. And certainly it's least concerned most of it on its European distribution. Still, it is listed as a Natura 2000 species, so protected at the EU level, and this protection is connected to Greece and some Greek populations. I will talk about this just a little bit later. So this is the distribution of the species. Actually, that was the best distribution map I could find on the internet. It's not the absolute best map, because this is based on individual observations, and certainly those countries where most observers are, those are overweighted. Nevertheless, the species does really have such kind of distribution, so it's quite common in the western part of Europe. We can actually say it's a nemoral species, so it's connected to temperate broad-leaved forests. And what is important for our subject, it is missing from most of the northern countries, Nordic countries, and it becomes quite rare in the boreal zone of Europe. It's also rather rare in the Mediterranean region, although it is still present there, and it's not as rare as in the Nordic countries. It's a typical European species that goes exactly to the geographic boundary of Europe in the Ural Mountains and in the Caucasus Mountains. Some occurrences exist in Asia Minor and the Near East. So, as I said, this is not a rare species. What is peculiar with this species and why it is listed as a Natura 2000 species, so protected by EU, is a peculiar population in Greece, in Rhodes, this island of Rhodes. And in this island, the adults have a very unusual behavior. During the height of summer, they aggregate in a special valley, and in this valley you can find them in millions of individuals, and after copulating here they lay eggs, and this behavior you cannot find in other parts of the distribution, although some literature mentions it from Asia Minor, so the nearby Turkish territories, the same behavior, but basically this is very unusual in Europe. And this very unusual behavior granted subspecies level recognition of this form in Rhodes, although nobody has ever found any genetic difference between the Rhodian population and the rest of Europe. Actually, it's very, very spectacular, the aggregation of the adult individuals. In this very special valley, they can be found in millions of number of millions, and what is also very unusual is really that it's a touristic interest right now. So a lot of people are coming to Rhodes only to see this very unusual event. Okay, so, but this is the reason for the EU level protection, this subspecies, which, as I said, and I would like to emphasize again, has not been found or studied, we don't know whether this is really a subspecies level differentiation. Okay, basically we have a frequent moth species, which is rather common in Europe, but we don't know, and yet it is protected, and we don't know anything about the genetic structure of this nature protected species. So what we have done before this study, it was one of my co-authors who did this very nice job. He collected all localized, so all barcode samples, cytochrome oxidase 1 mitochondrial DNA encoded gene samples, which had an exact locality on both systems. And what he did, he made a very preliminary picture, and what he found was very, very typical, I think. We have very little phylogenetic structure here. Most of the specimens belong to the same common haplotype, and there is one individual from UK, from Buckinghamshire, which is very genetically distant from the rest. We don't know whether this is only some sort of sequencing error or what is the reason. The other individuals included in this network, they are satellite haplotypes, and one more remote haplotype we can find from a Persian individual. But basically, and this is the most important message of this slide, the species displays very little mitochondrial barcode DNA diversity. And yeah, the Rhodian specimens, they all go into this middle or central haplotype, which of course contains most of the specimens. So this is our collection. So this is our collection sites. Here the dot size is corresponding to the number of individuals sampled. We sampled some populations in higher numbers because we wanted to do some basic population genetic diversity analysis. And then we also included a satellite population from Croatian coastland, and we also included these Rhodean population from the so-called Butterfly Valley of Rhodes. Okay, so this is our sampling on that map of distribution of the species. I must emphasize again that here we need to take this map with caution, because this is based on individual observations, and certainly it's overweighted by those countries where the observers are more numerous. So, but this is the sampling map in Europe. What we did was rod-seq analysis. It's a reduced representation genomic library approach that produces anonymous loci. Usually thousands of loci are sampled, and we are usually calling alleles from these datasets, and actually we are using the allelic frequency information and the allelic pattern, allele makeup, as source for population genetic and phylogenetic analysis. Okay, what we have exactly done, we multiplexed 72 samples, which included 3 technical replicates. We used the original rod approach, which is also called the single digest rod. And the advantage of this is we could bioinformatically filter out PCR duplicates. We did DNA Illumina sequencing of the rod-seq library. We obtained something like 632 million reads. That means on average 8.8 million reads per sample. It was 150 base pair paired end sequencing. We demultiplexed the rod-seq library samples using process rod tags. We are lucky, and this research is absolutely timely, because the chromosome level reference genome has just been published by the Sanger Institute in the UK. So we could work with a very nice reference genome of the species, and we used this genome to map the mitochondrial reads back to the mitochondrial genome and separated it from the nuclear genome, those reads which are coming from the nuclear genome. And these mitochondrial loci, we were super lucky that haphazardly, I need to say, rod-seq also sampled the cytochrome oxidase 1 locus, which is very unusual, I think, but we were lucky to do that. So we could merge our rod-seq dataset with that one which was published on BOLD system. So we evidently had a very nice cytochrome oxidase 1 dataset. We did some haplotype network building, we did some phylogenetic tree building based on these mitochondrial loci. Then we also did the nuclear reads. We mapped back them to the reference genome, marked duplicates, called the haplotypes and indels with free base. Then we made some variant filtering with VCF tools. And finally, we reconstructed a phylogeographic picture using IQ3 with all necessary, I hope, all necessary preparations or everything what is needed for genome-wide SNP analysis. Okay, and these are the results now. So, as I said already, very surprisingly and absolutely haphazardly, RADSEG sampled 2 mitochondrial loci. One was 16S RNA coding gene, something like 500 base pairs we could reconstruct, and we also sampled the cytochrome oxidase 1 gene. So this is the barcode gene, what all the butterfly researchers are using as a first standard. We managed to put together something like more than 1100 base pairs of this gene, which is quite a large bit. You can see it here in the lower part of this figure, and it was fully compatible with that part which we found in bold, because the barcoding region is always the 5' end of this gene. So the thing is, we managed to merge these data sets. I also included Callimorpha dominula, this species, as an outgroup. It is also mined from gene bank, the sequence of the whole gene sequence of this species, the outgroup. Okay, this is the result of the analysis. You can see here on the left side the TCS haplotype network. It's very similar to the picture what we have seen. This is the outgroup sample, which is the farthest from all other haplotypes. We still have this central haplotype, which contains 94 samples. Then we have this very, very far away haplotype, which originally we recovered from the UK in the bold system dataset, but we managed to add some more samples from different, and I need to say, random parts of the distribution area. Some are from Czech Republic. One is from the Butterfly Valley in Rhodes. Then there are these satellites from Germany, Czech Republic, also Germany, and somehow genetically closest to the outgroup we find the Persian sample, which was included from bold systems. The same very limited phylogenetic resolution goes for the phylogenetic tree. I'm sorry for the small text, but I will explain to you what are the important parts here. As you can see, most of the samples are unresolved in a very, very big polytomy, so we don't know anything about their phylogenetic relationships. What we can find is lower basal-most point is, of course, the outgroup Callimorpha dominula. Then the first basal lineage is represented by the Iranian specimen from Iran. Then we have one specimen from Germany, actually this one. And then we have the rest here in this very large polytomy. So basically, this is only what we can see. Actually, I believe this is absolutely normal for such a species which active spread and active expansion on its European area. I would expect such limited phylogenetic resolution, and most probably there is also a lot of incomplete lineage sorting going on. So evidently the barcoding region is not enough to understand the genetic structure. Okay, so let's move on and let's turn now to the RADSEG dataset, which is of course, which is where we expect some phylogenetic resolution. and we expect that because we obtained more than 300,000 SNPs and indels after the initial haplotype calling by freebase. But then I made a lot of filtering. I filtered for individual missingless, 10% individual missingless. I also filtered for biallelic SNPs. I removed all minor alleles which were lower than 5% frequency. I also filtered for minimum read depths of 5 reads. I also removed potential copy number variants. Those are the variants which are probably coming from paralogous loci. There were quite a lot, 37,000 such SNPs. And I also thinned the dataset to break down linkage between these SNPs. And after thinning for each SNPs must have been separated by at least 1,000 base pairs. We still had 42,000 SNPs, something like 42,000 SNPs. It's quite a lot. And this is how the picture looks. This is the IQ tree, a maximum likelihood tree, which was constructed using ascertainment correction bias, which is a prerequisite for genome-wide SNPs. And what is interesting here? Oh, okay, I need to tell we don't have an outgroup here, so this routing is on midpoint, and midpoint routing only recovered those parts of the tree as statistically supported, which are the basalmost lineages. And what are these basalmost lineages? Oh, I'm sorry. I also wanted to say first, here are the technical replicates. So the technical replicates are simply those samples which are repeated in the same library on a different point of the library, in a different sublibrary. And if they come together at the end of the analysis, it's a very good positive feedback that we done everything correctly, because certainly the same samples must come together at the end of the analysis. And as you can see, the 3 pairs of technical replicates came together, so it's okay, that's fine. Okay, now I would like to move on to the main message of this IQ tree, maximum likelihood tree. So what we recovered is the Rhodean samples in the root position. I need to say again, it is midpoint routing and not proper phylogenetic routing. Then the next lineage is the Croatian sample. And all the rest is a big mess, I need to say. So basically the rest of the samples are mixed up. There is no geographic structure within this phylogenetic tree. And I also need to confess there are in some cases significant support for association of samples, but I believe this is just a technical issue, because if you are working with 40,000 data points, random association can really happen, and because of this large number, some samples can come together significantly. Okay, this is the reason why I didn't put there those branches which are statistically significant, because I believe those are only technical significances. Okay, but the thing is, and this is the most important message, we don't really have any geographic structure within most of the samples. Okay, so this is how the picture looks on the map. So the Rhodean sample is very difficult, the Croatian sample is also very difficult, but the rest of the sampled area is a big mixture. Just to get some further information, what we did, we divided this sample distribution area into arbitrary geographic groups. So, for example, Eastern Romania, Western Romania, Southern Poland, Northern Hungary, Central Hungary, and so on. And with these geographical groups, we calculated Weir and Cockerham's genetic differentiation, because that was also some information. So the genetic differentiation overall is quite low, 0.08. It's overall an absolute value quite low. Nevertheless, the Rhodian samples are the ones which are the most different from the rest of the samples. Then we have here something very significant. All the Bohemian samples, all the Bohemian populations, so those are this point on in Western Czech Republic, those are very different from the rest of the samples, and now comes only the Croatian sample. Then here we really have very minute differentiation. So what does this tell to us? Evidently, this tells to us that this very northwestern part in the Czech Republic is really the leading edge. So this is the population, the part of the population where the populations have really recently expanded into. And this is the reason for the high level genetic differentiation, because these populations must be suffering from founder effect. The rest is not so structured, as you can see here, but that's probably the continuous area. So quite randomly, by restricting our sampling to our countries, the Visegrad countries, we found here a leading edge of the species. So based on our data, we can suppose the following colonization history. So evidently, probably some last ice age story or something like that, we must have had a southern refugium somewhere probably on the Balkans, and the species has colonized northwards. And this is what we can see from sampling this southeastern part of the distribution area. Unfortunately, we don't have a full picture because we have failed to sample Central and Western Europe. Okay, this is also something interesting. If you recall the cytochrome oxidase 1 very limited phylogenetic structure, we can suppose an additional movement to the west from the Persian region. So I can imagine that this is really the sign of a much older story, what shed light on the European colonization of this species. Okay, so, but this is the genetic picture. So on the sampled area, most of the sampled area, we have a rather homogeneous genetic structure, which is normal for the continuous area. And what we have found is nothing new in the literature, it's only new for this species, which nobody else has ever studied before. Because I think this is simply the classical example of the European Thermophilus biota, what has been written down long ago by the now classical work of Gottfried Hewitt. Several cases, the Mediterranean refugia were serving as the colonization source of the mainland of Europe. And what is also not unusual is this westward colonization and having some species which is widely distributed and very frequent in Western Europe, but the origin is not Western European. And for that we can say, this is a good example probably, Primula vulgaris, where we found the same sort of westward colonization from the Caucasus region, but the species is super frequent nowadays in Western Europe. Nevertheless, the origin is on the east. I think this is the same story here. And finally, I wanted to give you an example also from our previous research, where we have found no genetic structure on the continuous area. This is a super frequent, super widespread butterfly species, Polyomnatus icarus, and we studied the genetic diversity, a genetic structure from France to eastern Romania via Hungary, and there were no genetic structure with 16 microsatellites on this large area. It happens when some species are super frequent and they have a continuous distribution area. So in summary, we need to say we have the classical case of Southern European refugium and northward expansion. We can witness the active colonization pattern on one edge of the area, little genetic structure, and what I need to tell you is that we couldn't really find a conclusive evidence for the subspecies level separation, because although we can see a very stable separation in the genomic data set for the Rhodean samples, Rhodean population, I need to say, without a comprehensive picture on the whole area, I think we cannot say that we can confirm the taxonomic status of the Rhodean subspecies. Yes, so this is my presentation. Thank you very much for all those guys and ladies who provided samples, and we are also very grateful to the Visegrad Fund who provided us money for the laboratory work, and finally thank you for your attention.
Thank you, Gábor. And we have a lot of time for discussions, questions, and those sort of things. So please go ahead, Tamás.
Yeah, very nice talk, Gábor. I really enjoyed it. Now, one potential reason for the difference between the mitochondrial and nuclear distribution is the fact how they inherit it. Mitochondria only go down the female line. The autosome, the nuclear, should go on both sexes. Yeah. Now, I don't know much, but butterfly has ZW system, right? Is it the same here as ZW, like birds essentially?
Yes.
Yeah. So it means that you don't have sex-specific marker, right? Only autosomes.
We have, but we have not separated them, because we didn't map separately the sex-specific SNPs. All the SNPs what we were using were coming from all the autosomes.
I don't want to be cold, but you should separate it, because this can be a very interesting story. A number of years ago, we did a similar work with Kentish bladder. It was microsatellite based, and it turned out that the mitochondrial genes were a lot more homogeneous, a lot more panmagnetic than the autosomes, especially now the male, which is ZW, ZZ. So the males are the homogenic sex in butterflies. So, and the story was, and we actually repeated these samples with a large number of genomes, and it looks consistent, that females move a lot more than males, And this is the consequence of this, that the female genes move a lot more in the population than the male ones. Okay?
Yeah.
It may or may not be the same. Now,
Yeah,
this
absolutely.
is issue number one. Issue number two, I remember from Zoltan Varga, some of the butterflies and moths are monogamous, some others aren't. I don't know where the species belong to, but it would be interesting to compare the phylogeographic patterns, because in monogamous species, what we see, at least in some birds, the spatial segregation lot more characteristic, even microsatellites, than with polygamous one. And again, the reason is sexual selection, dispersal. In monogamous species, they grew up in one place, the offspring are side-graceful, males and females stay together. Polygamous, they distribute. So these two aspects, even if you just compare to other species, could give it a different spin. I really enjoyed this story, but if you're a scientist, it's the fact that you have money to do something is not a good reason. So I think you should really revamp it. At least one who is hook in English. You try to reason why. Of course, we all love it. This is the most wonderful species I ever seen in the world. I'm sure it is, but actually, you need to have some biological reason why you actually did it. Yeah. But I think it's a very interesting story.
Yeah. Thank you very much. Actually, these are very super useful advices. Thank you. I just need to say it was not me who picked these species. It was my Czech colleagues who are the lepidopterologists, and I think they were originally interested in these species because it's EU protected, yet we don't know anything about it. We suspected this would be the genetic structure. We suspected, but nobody ever done that. That was, I think, the reason why we picked this particular species. Nevertheless, those ideas, what you said, I think are very important, because especially if we have a chromosome level reference genome, why not to make use of it?
Yeah, you should.
Very good idea. Thank you.
Other questions? I will argue a bit of your conclusions that you are saying that the Czech populations are the leading, the rare edge, for instance, and the whole species spread from Rhodos to Europe. An alternative explanation might be that Rhodos is an island. So what you have seen there is basically just a founder effect. And if you cannot, so to say, falsify this hypothesis, then you cannot say that this is something happened here, what you said. And for that you would need much more sample from the Mediterranean on the things, because at the moment this is quite inconclusive, so it's quite a brave statement. Which is not really supported by the data, I think.
I must agree. I should have put this part a little bit more meaningful way or sensitive way. I'm not saying it's Rhodian origin, I'm just saying it must have a southern refugium.
Right.
Probably because the Croatian sample is also a southern sample. It's also representing the rear edge of the distribution.
Yeah, but that's on the seashore, which is quite separated by high mountains from the rest of the thing. So in a way, it's a kind of island population too.
Yes, this is something what we need to think through with those who know the biology of this species much better than me.
Definitely, it has quite a big dispersal possibility of this species that shows that we have no genetic structure in the northern part. And this is also supported that they just get together in one point in Rhodos and then spread all over the islands. So I'm not sure.
Where I must fully agree with you, and I wanted to emphasize this in the presentation, we don't have yet a comprehensive picture. I confined my statement therefore for judging the taxonomic status of the subspecies, but because we don't really have a comprehensive picture, it's just a kind of snapshot of this southern eastern part of the distribution area. I must agree with you, we cannot draw very firm conclusions. So what we can say is that our current evidence is hinting at such a story.
I wouldn't say that.
Yeah, I think it's the
two samples and other things which is very strange, instead leading edge in the middle of the distribution.
Well, in my view, if I'm not imagining it the right way, but simply from the Balkans, this animal was moving to the north, and that is leading edge in this respect. And unfortunately, it's really not a comprehensive picture, because we don't know what is in Western Europe. Probably there is another leading edge here, and like in several species, here we have a hybrid zone of different lineages, probably in the middle of Germany. Probably this is the reason why we have seen some cytochrome oxidase 1, very different cytochrome oxidase 1 haplotypes from Germany, United Kingdom, and so on. I don't know. Well, we don't have a comprehensive picture, it's true. It's a snapshot.
This is just a suggestion that you should also present at this stage of these alternative hypotheses.
Yeah.
Because
Okay.
it will be a more balanced picture. And one more question that have you any chance to figure out what part of the genome differ in rodents from the rest of the?
Yeah, this is what we have not discovered yet.
Interesting, because that might give a hint for this very strange mating behavior.
Yeah.
Is that the only place where they congregate? You mentioned your
Yeah. This is what I
Or
just.
is it the only place where buffalo or most people went?
Yeah. Exactly. This is the right way of saying that, because I have recently learned from Martin Konvička, one of the leading lepidopterologists of this project, that the Rhodean population is well known in the EU. But in Turkey, there are other populations which show this aggregation. So probably it's much more widespread. And actually, this is something what is a very good future direction to study this species. Because if we obtain samples from Turkish such population, and we have the reference genome, and we can do not RAT-seq, but we can do resequencing, that could really separate those genomic regions which are responsible for this aggregatory and unusual behavior compared to the rest of the area, of course. Probably it must have some evolutionary importance. For example, during the Ice Age refugium, that was a kind of behavior what was necessary, and it survived the different...
If you do full genome sequencing, you can reconstruct the population dynamic. Well, of course, I don't believe a word of it, but you can publish a paper how the population shrink and expand.
Yeah.
But anyway, so you can actually have thoughts that you can actually test a lot of these ideas.
I think it's a very good further direction for these studies. Hopefully, there will be somebody who will do this.